Corn. This book talks a lot about corn, basically saying, hey, there's a lot of people in the world, and there's going to be a lot more soon. So corn is a very popular crop for food. Meh, it's alright. And for energy. Corn oil, or ethanol, is a thing that's used for fuel. And we can design corn plants to be more oily, to yield more oil. It's just not as easy to do since corn oil production is controlled by more than one gene in different loci or in different. So how do we know which of the 32,000 genes are the ones that control quantity of corn oiliness? Well, scientists did as scientists do when they crossed a bunch of plants together. They started with a high oil plant and they crossed it with a low oil plant, and then a few crosses later, they managed to pinpoint the genes, the exact genes that make a corn oily. As it turns out, corn oiliness is controlled by 5 genes in chromosome 6. The karyotype of a corn only has 10 chromosomes, so that's not too bad. But there it is. Just as a refresher, something that is quantitative means that it measures how much of something, versus qualitative, which means that it measures or it describes the quality of something. For corn, for example, you can have a lot of oil, or you can have a little bit of oil, or you can have a medium amount of oil. That's the amount that's being described. Things like what our boy Mendel did are qualitative. A seed can be round or it can be wrinkly. There are no different degrees of how round a seed is. It either is round or it is not. I put a picture of a litter of puppies there because that shows another example of a quantitative trait. So a litter can have 1 puppy, or you can have 3 puppies, or you can have 5 puppies, but it's an amount of puppies. And I like puppies. Back to corn, however, scientists found that there's 5 genes controlling corn oiliness. DGAT1/2, I'm going to call it DGAT, is an enzyme that finishes prepping triacylglycerol, which is just a fatty acid. Plants that contain high oil levels have an extra codon of phenylalanine in the enzyme DGAT1, DGAT. This is what's making the corn plants extra oily. So scientists decided to add an extra codon onto some transgenic plants, which made it so that these transgenic plants produced more oil. The extra codon on these corn genes was not a random mutation. It is actually found in wild corn, so that means that at some point the corn must have lost this codon for whatever reason. I don't know, I hate plants. Plants are weird. But oily corn, that's great. It's great for ethanol and other stuff like that. But what's really, really cool, however, is that we have also been able to identify genes that can allow us to control the quantity of stuff certain plants make. For example, we can add vitamin A to rice, which makes this thing called golden rice. And we can also control the sugar in tomatoes, which I guess is great for ketchup. Quantitative traits are made up of many different genes in different loci. This leads to hella variation. Although both continuous and discontinuous traits work with the same set of rules established by Mendel, which is great, discontinuous genes are the ones that are easy peasy, lemon squeezy. It's straightforward. One genotype produces one phenotype, like rolling your tongue. You can either roll your tongue or you can't. Continuous genes are the ones that are stressy, depressy, lemon zesty, because one trait is made up of many different genes in different loci, which just means that they can be arranged in any random way when it comes to independent assortment. One of the three genes can be different, so in anaphase, when the genes separate, they don't have to stay all together. Skin color is a good example because it falls on a bell curve. Traits that can be measured with amounts fall in between two extremes, like the heights of the two men in the previous slide. Then you can have two people that are 1.73 meters tall, but if you look closer at their heights, one of them can be 1.735 meters, and the other can be 1.739 meters. This can go on for infinity. It's basically the same idea of asking how many numbers are in between 0 and 1. But because science is hard, there's different types of quantitative traits. You have the easier ones, which are the meristic ones that use whole numbers, like a cat that can have 5, 6, or 7 kittens, but she can't have 4.3 kittens. You can also have threshold traits, which you either have it or you don't. This isn't qualitative, even though it seems like it, but there's multiple factors, including the environment, that add up together until a certain threshold is passed. That is called susceptibility. If the threshold is passed, then you have the trait. If the factors influencing the trait don't pass the threshold, then you don't have it. The amount of susceptibility to a disease is the quantitative trait. Cleft palate, for example, is a birth defect caused by multiple factors. A little bit genetic, a little bit environmentally depending on what the mother was exposed to while she was pregnant, like smoking or certain medications or having diabetes before conceiving. Those are factors that can affect whether or not a child will be born with a cleft palate. This guy Nilsson had some wheat of different colors, and he made the wheat have sex with each other. He noticed that the pigment in wheat plants was controlled by 3 genes, but only 2 of those affected pigment, A and B. Having a plus means that you produce red pigment. Not having one just means that you produce no pigment at all. He picked 2 colors and bred them until they were homozygous, 1 white and 1 purple. He bred the white one with the purple one and got plants that were all red. That must mean that all the red plants are heterozygous. Then he interbred F2, the filial generation 2, when he got 5 colors of plants in different ratios. It's basically the same concept of a dihybrid cross, but A and B are not different traits. They're not different pigments. They both produce the pigment red, but they are additive. The more pluses you have on your genotype, the more doses of pigment a plant is going to have. So more pluses means that they're going to be darker. At least with the Nilsson-Eel cross, the environment didn't really play a huge part or a part at all in the influence pigment distribution had on the offspring of these two plants. So that's good, but that's always something to think about, that the environment can always mess something up. And here you can see what looks like kind of a dihybrid cross with the results. Just remember that it's not a dihybrid cross because both alleles A and B code for the same thing. It's just a matter of the dosages of pigment that you inherit. This is just an example of a problem that may arise, or a common genetic problem that involves the polygenic traits. It says, in the cross between a dark red plant and a light red plant, what are the chances of getting an offspring that is red? Well, it's asking about the phenotype, not the genotype. Not the genotype, which is good because in that case, the first thing that we need to do is identify the parents. I will choose a parent that is dark red with the genotype A+, B+, and B. And I will choose a light red parent with the genotype A+, B, and B. Now we need to find the offspring that are red, but to be red, you can have 3 phenotypes. It's those 3 that are right there. So since either of these 3 will give rise to red offspring, how many of the offspring in this cross will be red will have those phenotypes? So the third thing that we have to do is we have to find the ratio of each of the red genotypes in the parent cross, or in the cross that we're working on. First, we'll try the very first one, which is A+, B, B. You do a little Punnett square, just like in the dihybrid cross. You cross the first A of both the parents, and you figure out that in order to get A+, from the cross, it would happen half the time. And then you do the same thing with the B. You cross the B's of both parents, and in that case, BB would also happen half the time. So we use the multiplication rule to figure out how often that specific genotype is going to happen, and it's 1/2 times 1/2 equals 1/4. It'll happen 1/4 of the time. Then we try the second red genotype, the A, B+, B+. Turns out that we actually can't have this specific genotype with our original cross. It just can't happen because we can't have an AA allele from the parents that we were given. So this one doesn't count at all. Even if the B does work, the A doesn't work, so it doesn't matter. We discard it. We work on the third phenotype, which is A+, B+, B. That one's actually homozygous. And you check your little Punnett squares for A and for B, and A plus A is going to happen half the time, B plus B is going to happen half the time, so the whole genotype is going to happen 1/2 times 1/2 equals 1/4 of the time. So from the cross that we were given, we can only have 2 genotypes that give us red plants, 2 out of the 3. Those are the A+, B+, B, and A+, A, B+, B. So the original question was, what are the chances of that our cross will have offspring that are red with the 2 possible genotypes? I'm sorry, it says phenotypes right there. So we use the addition rule. For the first genotype, that happens 1/4 of the time. For the second genotype, that happens 1/4 of the time. We add it, and it gives us half. The answer in this case is going to be in a cross between a dark red and a light red plant. Half of the offspring will be red. This part is just extra. I like seeing Punnett squares because the letters can get confusing sometimes with the symbols. So what I did here is I took both genotypes of the parents and I made a dihybrid cross. And as you can see, the first genotype, the A plus A, B plus B, happens 1/4 of the time. And the other one, the homozygous one, the A plus B, B happens another 1/4 of the time. The question was asking how many of the offspring will be red. Those two genotypes give us red, so that is 8 out of 16, that's half. And that's when they're asking for phenotypes. But they can also ask for genotypes. They can ask from the same cross how many offspring will be red and heterozygous. Heterozygous being the different ones, the A plus A, B plus B, that happens 1/4 of the time. And the other question would be how many offspring will be red and homozygous, the A plus B, B, or AA, B plus B plus. We only have the first one, so that would happen another 1/4 of the time. That's just one of the questions that was hard last chapter because of the wording. It was just worded wrong because it was asking for a different ratio from the genotype and the phenotype, so that's just something to keep in mind. Here on this slide, we have the results of the Nielsen cross. You have the ratios, which are a little bit different from the dihybrid cross from the last chapter. But I made a little chart that has the phenotypes and the genotypes and how much doses of pigment each have. So, for example, you can have a dark red plant that has, but with two different phenotypes. And so on. This is in the book, so. At least the wheat plant only had 2 genes influencing the pigment. But since nothing is ever easy in genetics, statistics will be essential. Here are some concepts to know that are important. They are the distribution, the mean, and the variance. Frequency distribution tells us the number of phenotypes in a group of individuals and how they're arranged. For example, in the case of wheat, we had 5 phenotypes: purple, dark red, red, light red, and white. Frequency distribution will tell me how many of each of these individuals I have. So we use graphs. On the y-axis, you put the number of individuals in the group you're studying, and on the x-axis, you put the number of traits your individuals are showing. Mice are a good example to use to understand the different distribution frequency types. Mice live in the woods. There can be black mice and white mice and gray mice. These guys have many predators, including foxes, cats, and owls that share their habitat. The first kind of distribution is called the normal or bell-shaped. So these mice live in the woods, and they're black and they're white and they're gray, and they have these predators, but these predators are active during different times of the day. At night, black mice survive more, while the white mice get nommed on by owls. During the day, the white mice are all good, but the black mice are SOL, and they get nommed on by cats and foxes. Gray mice, however, are able to survive more, both during the day and the night, more than the two extremes. So evolution eventually, with time passes on, it does its thing, and the majority of the mice end up being gray. The second type is the skewed or directional frequency. So say for some reason all the owls all of a sudden disappeared. Since the night predators are gone, it's more convenient to be darker. So the curve shifts to favor one extreme. There's going to be less gray and less white mice as time goes by. The third kind is the bimodal, or diversifying. This is when the extremes happen with more frequency than the middle. Let's say that all the white mice all of a sudden began growing skin flaps between their front and back legs. Now they can get on trees and they can glide away from predators. The black mice, on the other hand, don't grow any flaps, so they can just scurry away and hide like normal. Gray mice then grow these half flaps, so they're not particularly good at gliding like the white ones are, but it also slows them down when running away from predators like the black ones do. So the pressure is put in the middle, since it is no longer convenient to be gray. As time passes on, these two modes on each extreme can become different species. First we have the mean, which is just basically the average. The book gives us that equation, if you want to memorize it or whatever, but it's very easy. All you gotta do is add up all the individual measurements together. So for example, I had these 5 boys right there. They range from 160 centimeters tall to 167 centimeters tall. So all of that gets added together. And then after all of that's added together, all you gotta do is divide it by the number of measurements that you had. So we had 5 measurements from 5 boys. That gives us 163.4 centimeters tall as the mean for this group of stick figures. The variance tells us how varied or how many kinds of measurements a group has. In the case of the mice, for example, there were only 3 traits. There was white, there was gray, and there was black. In the wheat, there was 5. And in the height example from the last slide, it was 5 types of measurements. So to get the variance, it's also easy. All you got to do is find the mean first, then take each measurement and subtract the mean from it, and then square it, and then you add it, and then you divide by this the number of measurements that you had minus 1, which I don't know, I thought it was arbitrary, but I'm not very good at math, so the end of result of that equation gives you the variance. The symbol for variance is s squared. Here we can see that from this specific cross that yielded 100 individuals, variance is shaped like a bell or a normal curve with a line going right through the middle of the peak being the mean. So number 3 is red. The mean color would be red. The last part of this section is about how this guy called Edward East, he studied tobacco plants, and he studied 2 types with different flower lengths. The first one had a mean length of 40.5 millimeters, and the second one was taller. It had a mean length of 93.3 millimeters. Then, after lots of interbreeding, we got some homozygous boys in every loci that contributed to flower length. They were all the same, all homozygous, one for tall, one for small, one for tall, one for short. Then he bred the 2 homozygous plants together, and he got medium-sized offspring in F1. Then he interbred F1 and got F2, but F2 ended up having a lot more variations than F1. So they didn't all have the same genotype, since flower length is an additive trait. So that's a lot of hoopla for just a couple of tobacco flower lengths, but what was the point of the experiment? When Edward East made his F3, he noted that the height of the progeny depended on the parents that were used. If he crossed two F2 plants, one of 50 millimeters and the other with 60 millimeters, the progeny of these two plants would have a mean of 55. If he crossed another set of F2 plants, one being 88 millimeters and the other one being 90 millimeters, the offspring would have a mean of 85 millimeters. All of this is just to say that flower length differences are partly genetic and they are passed down to the next generation. And that's it.